Hi guys, Miss Francis here to talk to you about our third and final type of receptor. This is a receptor that we already saw. It's the ligand-gated ion channel. They are protein pores that open or close in response to some sort of signal, thus allowing or blocking the ion flow. And what that's going to do is change the concentration of the ion inside and outside of the cell, as well as change the charge inside and outside of the cell. So think back to our unit on the neuron. Ligand-gated ion channels played a vital role when sending action potentials. Sodium needed to go in order to change the charge on the inside of our cell in order to propagate that action potential. Well, acetylcholine is the ligand that would bind to this ligand-gated ion channel in order to open it to enable that sodium to move into the axon. When we did the my dog is broken example, we looked at nitric oxide. Nitric oxide is a small, non-polar molecule, so it doesn't require a receptor in order to move into the cell's cytosol. So some signals, some ligands don't bind to the membrane, but they're actually able to get through the membrane. Other signal receptors get dissolved in the cytosol or the nucleus of the target cells. These signals pass through the plasma membrane. For example, hydrophobic hormones such as testosterone, estrogen, progesterone, which are all steroid or sex hormones, thyroid hormones of animals, and as I just mentioned already, nitric oxide, which is a gas that is a small size, allows it to slide between the membrane phospholipids. Testosterone, like other hormones, will travel through the blood and enter the cells throughout the body. Once in the cytosol, these hormones will bind and activate to receptor proteins. So for these types of ligands that can make it through the plasma membrane without the help of a receptor protein embedded in the membrane, their receptor proteins instead are found inside of the cell. And what these activated proteins now act as are transcription factors, which will turn on or turn off genes. And in the case of testosterone, it's going to turn on the genes that control male sex characteristics. So what these activated proteins act as is transcription factors, which control genes that are turned on, i.e., they're going to control the genes that are transcribed into messenger RNA. So some of these transcription factors are activators. They're going to increase the expression of that gene, while others are repressors, meaning they're going to decrease the expression of that gene. Where gene regulation explains why there are differences between organisms with similar genes. So even though I have 99.9% similar genes as someone else, the genes that are turned on in me versus turned on in someone else are extremely different. We mentioned CAMP when we were talking about the G protein coupled receptor. CAMP is an example of a second messenger. Well, what are these second messengers? Second messengers are small, non-protein, water-soluble molecules or ions, and they rapidly diffuse throughout the cell. Second messengers participate in pathways initiated by both the GPCRs and the tyrosine kinase receptors. So we already saw cyclic AMP or CAMP. Another one would be calcium, where many hormones trigger the formation of CAMP in their signal response. And as I said before, we looked at the example of epinephrine, which will trigger the formation of CAMP as a second messenger within their signal response. Other examples of second messenger responses include increases in calcium that might cause contraction of muscle cells. In plant cells, calcium trigger responses enable them to cope with environmental changes. So this calcium as a second messenger is also present in both G protein pathways and tyrosine kinase pathways. These are some great animations that show you those CAMP pathways. So now we're going to compare and contrast different types of signals. Signals or ligands can fall into two categories. They're either hydrophilic or hydrophobic. So hydrophilic signals are your peptide signals, your protein signals, whereas hydrophobic signals are your steroid signals or your lipid signals. Hydrophilic signals cannot cross the membrane. They are charged, they are polar, therefore they cannot cross the membrane. On the other hand, my hydrophobic ligands or signals, my steroid signals, are able to pass through the plasma membrane. They are able to pass through that lipid bilayer. So my hydrophilic signals or my hydrophilic ligands require a receptor that is found on the membrane. In other words, they require transmembrane proteins, such as my G protein coupled receptor, my tyrosine kinase receptor, or my ligand-gated ion channels. Where on the other hand, my hydrophobic signals, their receptors are intracellular, meaning that their protein receptors are found in the cytoplasm of the cell. What type of action would a hydrophilic signal have? Well, it stimulates a signal transduction pathway, often using those second messengers, CAMP and calcium. Whereas hydrophobic signals, their types of action include combinations of receptor and then the signal, where the signal acts as a transcription factor, which then turns genes on or off. Hydrophilic signals usually have a short duration of action, for example, measured in minutes or hours. For example, adrenaline or insulin, I want those responses to happen rather quickly. Whereas testosterone, a hydrophobic or a steroid signal or ligand, their duration is long, measured in weeks or years. Think about it. When testosterone is released in a male, he doesn't overnight all of a sudden start growing facial hair. It takes time. Some examples of hydrophilic signals are insulin, glucagon, epinephrine, adrenaline. Those are all peptide hormones. Furthermore, neurotransmitters are also examples of hydrophilic signals or ligands. Examples of your hydrophobic steroids include steroid hormones such as estrogen, testosterone, progesterone, thyroid hormones, and NO, nitric oxide.